Hey everyone, welcome to, I think, our last lecture for chapter 4. So now we're going to be talking about gram-positive bacteria. Gram-positive bacteria are also a large, diverse group. We classify them based on the amount of guanine and cytosine nucleotides in their genome. So if they have more than 50% guanine and cytosine, then they're called high G+C gram-positive bacteria. If they have less than 50%, they're called low G+C gram-positive bacteria. So based on guanine and cytosine. So the gram-positive bacteria that have high G+C are all put into a taxonomic group known as the Actinobacteria. Most of these are soil bacteria, but some are aquatic. There are multiple genuses in this group. We're not going to look at all of the genuses. We'll look at a few of them though. So Actinomycetes, you can see right here, is I'll say a curved rod-shaped bacterial species found in soil. We have Corynebacterium here, and you can see that they oftentimes make this, they're also rod-shaped, but they often form what looks like a V shape, and that's known as a palisades. And then there's Gardnerella. Gardnerella have a unique shape, and they are pathogens that can cause disease. Specifically, this is showing Gardnerella vaginosis, which causes bacterial vaginosis. But there are other groups as well, Micrococcus and Mycobacterium. Mycobacterium and Micrococcus, as well as Corynebacterium, are all found in our lab. So some of these organisms are used in industry, while others are considered pathogens. So two of the major pathogens that we discuss are Mycobacterium that cause tuberculosis and leprosy. In our lab, we have Mycobacterium smegmatis that does not cause these diseases. So here you can see Mycobacterium from your textbook. This is figure 4.23. This is Mycobacterium grows in kind of a, it looks kind of moist there. I would say typically on our TSA agar, this is not TSA agar, it looks very dry and flaky. And then Corynebacterium diphtheriae causes diphtheria. Diphtheria is an upper respiratory infection that causes severe sore throat. It is often found or identified by the pseudomembrane that is formed on the back of the throat, causes problems with swallowing and breathing as well. So it's a very serious disorder, especially in children. And then we go to the low G+C gram-positive bacteria. The first group of our low G+C gram-positives are Clostridia. These are in general obligate anaerobes. They're found in soils, and they can form endospores. So there are pathogens in this genus. Clostridium perfringens produces a gas green. Clostridium tetani produces tetanus. Clostridium botulinum produces botulism. And Clostridium difficile produces C. diff infection or severe colitis. And that's what you're seeing here, are Clostridium difficile. And this is figure 4.19. Then we have the lactobacillales, which contain both bacillus-shaped species as well as coccus-shaped organisms. So just because it says bacillales does not mean they're all going to be bacillus-shaped. These include your Enterococcus, Lactobacilli, Lactococcus, Leuconostoc, and Streptococcus. In our lab, we have Enterococcus and Lactococcus. Streptococcus are are then separated into different serotypes by their ability to lyse blood cells. So there are 3 forms of blood lysing that can occur. We have alpha, beta, and gamma. So your pathogenic streptococcus, like Streptococcus pyogenes, is a beta hemolytic. So beta hemolysis means that it fully lyses the red blood cells, and that's why you're seeing these large opacities around the blood agar, because the blood has been broken down completely. Beta or alpha hemolysis is partial breakdown of the blood. Gamma hemolysis is no breakdown, so it does not lyse blood at all. And this is a beautiful picture of Streptococcus growing. So it grows in long chains of cocci. Enterococcus and lactococcus also form chains. They typically aren't always this long. A lot of times they're only about 3 to 5 cells in length. Then we have the bacilli. And the bacilli also include coccus and bacillus bacteria. The names kind of make you think that you're going to only have one type, but don't let the name fool you. So the two groups of organisms under the bacilli are the bacillus organisms and your staphylococcus organisms. So Bacillus anthracis is an example of bacillus species. Bacillus cereus is the species that we have in our lab. These are also endospore formers. And here you can see this is a gram stain actually of both E. coli and bacillus, but the bacillus are these long purple chains. They are endospore formers, so if you do an endospore stain on these guys, you'll find little endospores inside the cells. This is how bacillus grows. It grows in it typically will cover the entire plate at some point. It grows very quickly. So if you plate it in the morning at 8 o'clock, by 4 o'clock in the afternoon, you'll already see the cells, or not cells, but you'll see the colonies. And then Staphylococcus aureus, which is over here. You see Staphylococcus aureus grows in grape-like clusters. That's where it gets its name. And there are two major types of Staphylococcus or of staphylococci. We have Staphylococcus epidermidis and Staphylococcus aureus. So this is Staphylococcus aureus here, but Staphylococcus epidermidis looks the same. Staph epidermidis in general does not cause disease. It is found on our skin, and it is part of our normal flora. Staphylococcus aureus can be disease-causing, causes things like boils, can cause impetigo and other diseases, and can be hard to treat because of antibiotic resistance. The last of our low G+C gram-positive bacteria is Mycoplasma, which is a very hard organism to place. The reason for this is gram-positive bacteria. One of the major characteristics of gram-positive bacteria is the thick peptidoglycan in the cell wall, but Mycoplasma lack the cell wall. So I always say that bacteria in general have a cell wall. Well, this is the example. This is the rule breaker. Mycoplasma does not contain a cell wall. Because of this, Mycoplasma is called a pleomorphic cell. So it doesn't have one shape. It can look like a rod, it can look coccus, it can make other shapes, it can be star-shaped. You never know what it'll look like. It's not going to have one single type of shape, since it's just got that plasma membrane that surrounds it. Mycoplasma pneumonia is one of the species that fall under the mycoplasma. And this species causes atypical pneumonia, which is typically a less dangerous form of pneumonia. I also like to mention, since mycoplasma has no cell wall, if we do a Gram stain on it, what would it look like? I'm not going to answer that. I want you to think about that. If we do a Gram stain on mycoplasma, what is the outcome going to look like? We know it's a low G+C Gram-positive bacteria, so think about that, okay? And then you can look at page 181 and 182 for table 4.10, which summarizes all of your Gram-positives, low G+C organisms. The next group is the deeply branching bacteria. I only have a few more slides, so I'm going to finish this up on this lecture, which will only take about 15 minutes. So deeply branching bacteria are named because of their location on the phylogenetic tree. So here is the last universal common ancestor. And if we look, deeply branching bacteria are one of the most ancient bacteria around, okay? These organisms thrive in extreme environments, and many of them can live in conditions that were on Earth when Earth began. So one of these organisms, Deinococcus radiodurans, is also known as Corynebacterium. And the reason for this is this bacterial species can thrive in multiple harsh environments. So in drought conditions, it can thrive. Hold on. It grows in extreme heat. It can withstand ionizing radiation, and it can grow in extreme acidic environments. So that's really unique because most organisms might be able to withstand heat or withstand the acid or drought, but not all these different environments. And one of the reasons that this organism is so thrivable is that it has multiple DNA repair mechanisms. So Deinococcus radiodurans or Corynebacterium. The last group then is the Archaea. So remember, Archaea and bacteria do differ. They differ in genetics. They differ in ecology. They differ in biochemistry. The two characteristics that I've talked about that separate bacteria from Archaea is that Archaea have ether-linked plasma membranes, whereas bacteria have ester-linked plasma membranes. Bacteria have peptidoglycan. Archaea do not have peptidoglycan. So Archaea can live in all environments, just like bacteria. Some are called extremophiles. So they live in extreme environments, either high or low temps, maybe extreme salinity, extreme acid. None are known to cause disease. So Archaea are not disease-causing organisms. There are I think it says 5 phyla that they talk about, but your book really only focuses on 2 of the phyla, the Crenarchaeota and the Euryarchaeota. So those are the two phyla that I'm going to talk about, okay? Crenarchaeota are all aquatic. Most are called hyperthermophiles, which means they live in extreme high temperatures. They can live at the hydrothermal vents. One example is Sulfolobus. Sulfolobus is an organism that is a hyperthermophile, and it also is acid resistant. So it produces proteins known as aphids that are both thermostable and acid resistant. Aphids are used then to neutralize different antigens, and antigens are proteins that elicit immune responses. So Sulfolobus is important to us in biotechnology. Then we have the Euryarchaeota. One of the Euryarchaeota are your methanogens. Methanogens are able to reduce carbon dioxide to methane. Methane gas is what is released when cows fart. That would be an example. Halobacteria then are another example of Euryarchaeota. And these halobacteria require high concentrations of salt to survive. They live in up to 36% salt concentrations. They live in the Dead Sea. And what's really cool is they utilize photosynthesis. Whoa, I spelled that wrong. I didn't spell it wrong, I just forgot the space. So they utilize photosynthesis, and they use a protein called bacteriorhodopsin when they photosynthesize, and that actually causes the water to turn purple. So it's a really pretty color. And so these are water baths, salt ponds, that's what they're called. And they're purple because of the halobacteria photosynthesizing. That is all I have for you in this chapter. So it was kind of a long chapter, but not near as long as chapter 3. And I hope you enjoyed it and learned a lot about all the different prokaryotic organisms. Bye.